Hello. Okay, what I'm going through today is cell biology. So, gonna go through cells, mitosis, which is cell division, stem cells, specialized cells, osmosis, diffusion, and exchange surface. Different exchange surfaces. Tomorrow, at some point, I don't know what time yet, I'll put that up at some point, but I'm going to go through the heart, the lungs, circulation, and all of that kind of stuff tomorrow. But today, I'm focused on cells and specialized cells, osmosis, diffusion, mitosis, stem cells, that kind of stuff. Okay, let's get into it. Right. So, okay. Hi, George. Just waiting to see if anyone else is in the chat. Hold on 1 second, 1 second. We will start in a minute. If you wanna, hi Case. Yeah, if you wanna chat, you have to subscribe or something. I think so. We'll get started. So I don't know if I sent the link out to lots of people, but there we go. It don't matter. Yeah, so gonna go through cells to start with. So yeah, you can always share the link to other people if you want, so they can come on and do a bit of revision for their biology as well on Tuesday. First thing, what are the 5 labels there that are on this diagram of the cell? So see if you can put up on the chat the 5 labels. Okay, so just here we go. Right, let's get into it. Okay, so what are these arrows pointing to? See if you can put up the answers in the chat. This is an animal cell. Yeah, you got the mitochondria, cell membrane, ribosomes, yeah, cytoplasm. Nice. So they're all your standard 5 things. Brilliant. Okay, just a couple of extra questions. Well, do you know what each one does? So, what does the nucleus do? You just need to know one thing about each. Yeah, cytoplasm's a jelly. The nucleus stores genetic information. Nice. Do you know what the cell membrane does? What about the cell membrane? You could be asked about what the cell membrane does. Yeah, that's right, Case. It controls what goes in and out. What about the ribosomes? Or ribosomes or ribosomes? What do they do? Anyone know what the ribosomes do? Or ribosomes? Protein synthesis. That's right, George. They're for making proteins. And can you think of 3 things in the body that are made of protein? What kind of things are made of protein in our body? So these are the type of things that could be made on the ribosome. So they might not directly ask you where are proteins made. They might say something sneaky like where are enzymes made, and then you have to go, right, enzymes are proteins, they're made on the ribosome. So you've got to watch out for them sneaky questions. Okay, so you've got things like enzymes, antibodies and hormones. Those 3 things are all made of protein, and so they can all be made on the ribosomes. Anything made of protein can be made on the ribosome. And do you know which of these organelles is the biggest? Which structure is the biggest in the cell? And what structure do you think is the smallest? What's the biggest organelle? What's the smallest organelle? Mitochondria is pretty small, Sophie. Yeah, mitochondria is very small. It's the second smallest structure. There's one smaller than the mitochondria. Just going to sip my tea. Nucleus is the biggest case, that's right. Nucleus is the biggest. Mitochondria is small, but the ribosomes are even smaller. Okay, so the ribosomes are the smallest. This means that both the mitochondria and the ribosome you can't see with a light microscope. Okay, you need that special microscope to see those. They're so small. Okay, let's move on. Alright, so here it goes through what they do. The nucleus contains DNA, is in charge of cell activity. The cell membrane controls what goes in, what goes out. Cytoplasm, where chemical reactions happen. The mitochondria, aerobic respiration, not anaerobic, as we discussed yesterday. That happens elsewhere. The ribosomes are the smallest. They're the little black circles. So whenever you're looking at a cell, the smallest thing in there is going to be ribosomes. Okay, and that's where proteins are made. Okay, you know what's coming next? Standard. What are the 3 things found in the plant cell that you don't get inside? So what 3 things are found in plant cells that you don't get inside animal cells? Anyone got an idea? Vacuole, nice. Nice one, Alex. Cell wall, chloroplast. Yep, cell wall case, chloroplast, vacuole, cell wall. Oh, good, you've covered them all. Chloroplast, cell wall, vacuole. Does anyone know what chemical is found in the cell wall that gives it its strength? There's like an ingredient in the cell wall that makes it strong. Begins with a C. Cellulose. Well done, Alex. Yeah, there's a chemical in the cell wall. Now, when you go to the toilet, if you've had some sweet corn, you'll see the yellow piece. Not that you should be looking at your poo, but the yellow bits of sweet corn and that. They're the bits that humans can't digest. We don't have the enzyme cellulase that cows have that can digest the cell wall of plant cells. So we're not very good at digesting the cell walls, and we call that fiber. That's what just goes straight through us. That's why it's good to eat plants. They go through you and clean you out because we can't digest their cell walls. We can digest all the other bits, the cytoplasm, and we can take all of their starch and glucose, but we can't get rid of the cellulose. Right, perfecto. Of course, chloroplasts are the structure. We covered that yesterday. They contain the green chemical chlorophyll that absorbs light for photosynthesis. And then there's a vacuole which contains cell sap, water, sugars, minerals, nutrients. Okay, the cell wall is you'll just get the marks for saying it's for strength or protecting the cell. Better is it gives the plant cell strength, or it holds it together. It actually stops it from bursting and exploding when it takes on water. Right, now, we can split all cells on the whole of the Earth. Every single cell on the Earth we can split into 2 camps. One, which have a nucleus. So fungi, plants, animals, and protoctists or protists all have a nucleus. They have a special name. And then there's one other group for the bacteria that don't have a nucleus. Do you know the name? what do we call cells that have a nucleus? The answer is eukaryotic. So cells that have a nucleus, well done, are eukaryotic. So you need to know that. Um, do you know what we call cells without a nucleus? So we call cells with a nucleus eukaryotes. It means complicated. I don't know what language it is, whether it's Greek or Latin, but it means complicated cells. And we call other cells without a nucleus prokaryotes. Well done, Sophie. Well done, everyone. Yeah, prokaryotes. Now, this is, you only have to know about bacteria as a prokaryote. They're the only type of prokaryote. It gets more complicated. They're split into different types of bacteria later on. But you need to be able to label the structure. So I've got three things here that you should label. There's a similarity with plant cells, and then there's two things that are different. Now, what do all cells have in common? Every single cell has ribosomes, whether it's fungi, plant, yeast, whatever it is, animal, bacteria. They all have ribosomes. They all have cytoplasm. But not all cells have chloroplasts, mitochondria, or cell walls. Animal cells don't have a cell wall. Bacteria cells do have a cell wall, but what do we call the DNA in the middle? And what do we call the small rings of DNA? Anyone know what we call the loose DNA in the middle? And then we've got rings of DNA. Yeah, they are like spools of DNA. The little rings are called plasmids. Have you heard of plasmids? They're the small circles. Yeah, flagellum is the tail. Nucleoid, you can call it, or you can call it, some people call the shoelace or spool of DNA in the middle loop of DNA. They'll accept a lot of things. They'll accept you calling it free DNA or loose DNA or free floating DNA. So there's lots of things they'll accept there as the answer. Yeah, flagella is the tail. Some bacteria have that. And these little hairs coming out the side, you don't need to know their names, but they'll accept it in an exam if you were to write it. They're called pili. You don't need to know that. P-I-L-L-I, like pili. They're little hairs that stick out the side. You don't need to know that, but just in case you were wondering, here are the answers for those bits. So you could be asked, what is the similarity between a plant cell and a bacteria? And they both have a cell wall. They might say, what's something plants and bacteria have and animals don't? And that would be a cell wall. If they said, what's something they all have? You could say cytoplasm. Or cell membrane. All cells have a cell membrane. Right. Quick questions. See if you can have a go at these. These are to test your knowledge. You can just do them in your head, or you can type that. You can put the answers on the chat if you want. Which cell structure, if you put it on the chat, just put the number with your answer. So you know, like, oh, 1. What cell structure contains DNA? Well, what cell structure is where protein synthesis happens? What's the name for what do we call cell structures? Which cell structure is where energy is released? Name a cell structure found in plants, but not bacteria. Oh, plants and bacteria, but not animals, sorry. Name 3 cell structures found in plant cells. What is a plasmid in? Which of these 3 cells we looked at is the smallest? What name is given to the cells that have no nucleus? Yeah. Yeah, bacteria is the smallest, Sophie. That's right. So for number 8, bacteria is the smallest. A bacteria is actually the same size as a chloroplast or a mitochondria. So when you're looking at cells and you think of an animal cell or a plant cell and you draw the chloroplast or mitochondria, that's the size of a bacteria. Once upon a time, mitochondria and chloroplasts were bacteria that came to live in other cells. So they're a lot smaller. They're like 1,000 times smaller than an animal or plant cell. What's the green chemical, number 10, found in the chloroplast? And what about the structure found in plants and bacteria, but not animals? Yeah, chlorophyll's right for number 10, Sophie. Well done. What cell structure would you find in a plant and an animal, but not a bacteria? Yeah, cell wall, George. That's right. Cell wall are in. Sorry, they're in plant and bacteria, but not animal. Keep wording that the wrong way. And what's the structure where energy is released? What's the energy releasing structure of the cell? What one's where we release our energy? Yeah, mitochondria. Good, good, good. I'm going to put the answers up now, guys. You can just check them off in your head. Yeah, that's right, Sophie. Every cell that's known on the Earth, so far we've only found cells on the Earth, as we haven't discovered aliens yet. Every cell on the Earth has a cell membrane and a cytoplasm. So if you're ever asked to do a comparison, that's a really clear area to do comparisons. Cytoplasm and cell membrane are found in all cells. Ribosomes are found in most cells. All cells have to make protein. Another quick one, viruses are not cells. So if you're ever asked about viruses, they have nothing that a cell has. They have no cell membrane, no cytoplasm, no cell wall, no nucleus. They have none of the organelles. And we call the structures cell structures or organelles. So yeah, all cells have those, but the others, there are some differences between them. Okay, let's zing on. So these are the answers. Unless there is always possible that I've put the wrong answer somewhere. No, they look all right. Chloroplast, cell wall, vacuole, 3 things found in a plant. Small circle of DNA in bacteria cells is a plasmid. Bacteria is the smallest. Prokaryotes, things that have no nucleus or bacteria. And chlorophyll is the green chemical. Coolio. Right, let's move on. All right, this is an exam question. What you'll see I've done here, so I've used a highlighter. I recommend bringing a highlighter in. It will help with your breathing and staying calm if you're like in a panic when you hit a question. Just use the highlighter and take 10 seconds to go through and highlight the key points. You might miss something like this last bit I've highlighted, 2 significant figures. You might miss something otherwise. That's really key and you'll cost you a mark. All right, see if you can do these questions, guys. So the first one is name structure X. That's really hard. This is the type of one they're going to try and throw you off with. So it's this is a diagram of one cell. It's zoomed in so you can hardly see anything. This is the type of thing they'll do to you. So you've got to use a bit of common sense and say, well, I can see the ribosomes and the mitochondria. So what's this circle thing here that's structure X? And then I want to look at the top. I forgot to highlight. It shows a liver cell. So before we go, that big circle that we've got the corner of, it's a vacuole. Well, before we say it's a vacuole, this is a liver cell. It's an animal cell. So it can't be a vacuole from a plant. Okay. Hold on. Yeah, it's the nucleus. Yeah, it is a nucleus. That's right. Sorry, I got my kids screaming in the background like nutcases. Yeah, so X is nucleus. Function of mitochondria, that's 2 marks. So why would you get 2 marks for mitochondria? What should you guys be putting there for the mitochondria function? So, yeah, aerobic respiration to get you 1 mark, Sophie. That's good. You're right. Mitochondria is for aerobic respiration. What's the second mark? What does aerobic respiration do? Anyone got an idea? What is energy? Well done, Alex. And make sure you say release energy. If you say make energy, they won't give you the mark. Okay. Right. This next question is a magnification question. Does anyone remember the calculation or the formula for magnification? They'll probably give it to you in a triangle, or they'll actually give you the formula. So it's got actual size, image size. Some people call it real size. They've called it real length here. And then magnification all in there. Yeah, that's it. Image divided by actual. or you can say image divided by real gives you the magnification. So they've said that they've given us the magnification is 650,000. They've said the image was 27 millimeters. What's the real length? So anyone do the maths there? So the mitochondria, yeah, Sophie, going back to the mitochondria question, the mitochondria does aerobic respiration, and aerobic respiration is the process where energy is released. So the mitochondria, yes, it's where aerobic respiration happens and it releases energy via aerobic respiration. A bit like the chloroplast takes in sunlight in a plant cell and does photosynthesis. So the chloroplast is where photosynthesis happens, but and absorbs sunlight. So if they ask you what a chloroplast does, that's 2 marks, and what a mitochondria does is 2 marks. What about this maths? We've got the real length is 27 millimeters. The mag is 650,000. Sorry, the image length is 27 millimeters. The mag is 650,000. What would be the actual size? So you bust out your calculator here, which I shall show you now. So you get your calculator out, and that's highly crusty. So image is 27 millimeters, and we'd press divide by the magnification, which is 650,000, because image over magnification is going to equal the real size. And the real size, press SD to put it into yeah, I just messed that up. Right. That's the real size in millimeters. So a cell is very, very small. You can see there's 5 zeros there, so that's like 4.1 or 4.2 times 10 to the minus 5, or you could just write that number down. That's how big the real cell is. So it's very small. They want 2 significant figures, so I've messed that up if I don't listen to that. So they want it as 4.2 or 4.1. Let's have a look. 4.2 times 10 to the minus 5 is the answer there. So you go real image size over the real size of the object, and you got the magnification. So we just do 27 divided by 650. We get that. Put it to 2 sig. We don't include the zeros as significant there, so they go afterwards. But there we go. Right. Neat. Yeah. Right answer, guys. Yeah. And that's in millimeters. Now, don't forget, sometimes they're going to be brutal and ask you to change it to micrometers. If they want it in micrometers, you're going down a level from millimeters, you times 1,000. To convert millimeters into micro, when you're going smaller, you multiply by 1,000. When you're going back up from micro to milli, you divide by 1,000. So a little bit of a weird one. Right, guys. Moving on. I don't know if it's buffering because it's like a bit slow to... Here we go. Okay, cool. Thanks, Alex. Right. You got 2 microscopes. Light microscope, electron microscope. So, for AQA, OCR, and EdXL normal science GCSEs, you need to know how to compare the electron microscope with the light microscope. For IGCSE, you don't need to be able to compare the electron and light microscope here. But obviously, the ability of microscopes to make an image larger, what do we call that? Name an organelle you could see with a light microscope. Name an organelle you couldn't see, i.e. they're asking you what's the smallest thing that you can't see with a school microscope. And then what are the 2 advantages of the electron microscope? Yeah, so You can see a nucleus. Yes, it's the biggest one. So absolutely, you could see the nucleus. Yeah, you couldn't see chromosomes, Sophie. So you'd have to name, like, you can't see the chromosomes or the DNA that's in the nucleus. Or you can say you can't see ribosomes, the little black dots that make protein. You probably can't see mitochondria. You could see a plant cell and you could see the nucleus, Georgette, inside the plant cell, but you wouldn't be able to see the mitochondria or the ribosomes with a light microscope. Yeah. So the best thing about the electron microscope is it does have a higher magnification, but the best thing and most likely thing they'll ask you, it has a higher resolving power or higher resolution. So you can see all of the different structures and in more detail. And all you need to know is in more detail. You can see structures more clearly or more detail. There are other definitions, like it's the resolution is the ability to distinguish clearly between two objects or see two separate objects, but you don't need to say that. You can just say it's the ability to see things clearly. So that's the resolution. All right, let's zing. So you can see there, these are the answers. You can see the cell wall, the nucleus, the cytoplasm, the cell membrane. You're not going to see plasmids, ribosomes, chromosomes in the nucleus. The ability to see details more clearly is the easier one to remember than the ability to distinguish between two objects or see them as separate. Okay, they cost about £200,000, so your school won't have one. If you're looking at your school for an electron microscope, they ain't got one. They're a lot of money. Right. Now this bit you don't need for IGCSE, so the next 5 minutes you don't need this if you're doing international GCSE. This next 5 minutes, if you're doing AQA, EdXL or OCR on Tuesday, this can come up to your microscopes practical. Okay, so if you're just doing standard GCSE, biology or combined science, you do need to know this. So you could be asked how to prepare a slide, about 4 marks. Probably you'll get 2 or 3 marks max on it. You could be asked how to use a microscope to view the slide, and then the final one is how would you draw, which they've not really asked. I think they've like asked 1 or 2 marks over the years on that, so it's not been a big thing. But this core practical could come up. Remember, this is for all of the examples. I know it says AQA here. This is for OCR Gateway, EdXL GCSE, and AQA GCSE. 15% is going to be core practicals. On Tuesday, 15% of your exam. So if your exam is worth 70 marks, you're looking at quite a lot of them, almost like 10 marks are going to be on core practicals. So, yeah, there's only the osmosis, the microscopes, enzyme practical, and the photosynthesis. So there's 4 ones they can possibly ask you on. And if you're doing triple biology, they can ask you the culturing the bacteria. Okay, so how do you prepare a slide? Anyone know a couple of things about how to prepare a slide? So if they ask you how would you prepare a sample, what would you say? Oops. Okay, I don't know. Am I buffering a little bit, or is it seems to be going a bit slow? Yeah, you're right, Sophie. You don't want any gas bubbles. So if you take a glass slide, what do you put on the middle of it? Yeah, hold it on the sides. Okay. What do you put on the middle of the glass slide? Can you see the picture here of the slide, or is it on the answers still to the microscope questions? So I'm not sure if my... Okay, coolio. Right, so you need to place a water drop on the middle of the slide. Do you cut a thick bit of your sample or a thin bit of your sample? You can call it a sample or a specimen. Do you cut a thick bit or a thin bit? So if I'm looking at an onion, should I cut a thick bit or a thin? Thin. Brilliant, George. Why has it got to be thin? Why mustn't I cut a thick sample of tissue? If you wanted to look at my skin under a microscope, why would I need to cut a very thin slice and not a thick bit? Think about the name of the microscope. It's a light microscope. Yeah, it's to see the cells clearly. The light has to be able to get through them. So the microscopes we use in school, they're not cheap. They're still like £100. Yeah, that's right. So we can see the cells better. They've got to be thin so the light can go through. Now, what do I need to add? If you look at an onion and you look at the cells, it's just, you're not going to be able to see anything. I need to add something to colour all the organelles, to colour the cell wall. What do I add a drop of? Does anyone know what you add to give colour to all the structures? Yeah, so you can see the cells better, but what do we add? Iodine, George. Yeah, you can call it stain. Okay, in all the examples you can just say stain. There's lots of different ones. There's one called methyl blue or methylene blue. There's iodine. These will colour the structures and allow you to see the stuff better. Okay. And then finally, we lower, you probably used it in school, a little square of glass. It's like a little square. And you push it down. It's very hard to see. You lose them on the table at the end of the lesson and they snap in your hands really easy. Does anyone know what they're called? The little tiny square of glass. The square of glass is called a cover slip. So guys, you need to know, this is the 4 points you need to know. If you're asked about how to prepare a slide on Tuesday, drop of water, float a thin section on it, so cut a thin section. They might ask you why thin. Why did Sally cut a thin section? So the light can go through. Add a stain. Why did Raj add a stain to the sample? Oh, so we can see the details more clearly, or the structures more clearly. And then why did we lower the cover slip, the little bit of glass, push it down and give it a gentle push to remove the air bubbles? Why not squish it down? You don't want to squash the cells and break open the cell membrane, so you just gently push them down to remove air bubbles. Coolio. Right guys, you'll need to know how to label this. Okay, yeah, water is to give the cells something to float on. Sophie, so it's just really, if you're asked why put water there, it's just so the cells can float on the water. Yeah, you need to know all the, if they're going to ask microscopes, you need to know all the names of the structures. The main ones, the main structures of the microscope you need to know. You do need to know the coarse knob, and then there's a smaller knob called the fine focus. So there's a coarse or rough knob, which you turn, which brings the stage up and down. And then there's a fine focus to allow you to see things clearly. What you look through is the eyepiece lens, and then you have an objective lens, which is the one that you can rotate like that. And then the stage, which they've never asked about the stage, but the stage is the platform that you put everything on. Okay, I'm going to put up the, these are the labels, so you do need to know these for Monday, guys. Coarse knob moves the stage up and down. You look down the eyepiece, you raise the stage up, you put your sample on, and then you slowly turn the stage back down away from the objective lens, and then you can adjust the power and you can adjust the fine focus to make it really sharp, the image. Don't worry about that. That's like the order. I've kind of talked through it, so I'm just going to put the correct order. This is the correct order. If you were, so you could be asked like something like, Tom was looking at he made a slide with specimen of onion on it. He's prepared his slide. Now explain for 4 marks how he would observe the cells it using his microscope. So you could say clip it on a stage, 1 mark, select the lowest power, use the coarse adjustment knob to raise the stage, look down the eyepiece would be a mark, just if you're mentioning the eyepiece, adjust the fine focus to make the image clear, and then select the magnification you want, because you can then, once you've got it in focus, you can then change the magnification and pick a more powerful one to make the cell bigger, the cells bigger. Okay, so you need to know that in case you're asked that as well. Remember, 10 marks are on core practicals. So are they really going to ask 10 marks just on microscopes or just on osmosis? Or yesterday we did the photosynthesis practical. Are they really going to ask 10 on that? Probably not. You're probably going to get a combination of 2 practicals spread out through the exam. If you're doing the triple, if you're doing like GCSE biology, you're going to get more marks because it's more marks overall. So you're going to get, like, instead of 10 marks, it's going to be 15 marks on core practicals. So this is another thing. This hasn't really been tested. It's only been like 1 markers. They've not tested like 3 or 4 marks on this before. So you never know. Tuesday could be the day. I don't think it is, but just it could be. They say if you're going to do a microscope drawing, use a sharp pencil, clear unbroken lines, label the structures with straight uncrossed lines, and write the title and the magnification. So there's a mistake in all 3 of these drawings. Can you spot the mistake? What's the mistake in all 3 of these microscope drawings? Anyone spot the mistake in these 3 microscope drawings? There's a different mistake in each microscope drawing. Okay. What it's to do with the arrows on the sperm cell. It's to do with the arrows. Yeah, what it is, it's confusing because they've crossed the lines. Well, you got to write there the sperm cell. You need to keep your arrows separate. If you draw the arrows crossed, that's not allowed. Okay, so their arrows are going over each other so that it's hard to tell what's going on. Good, you spotted that one. What about in the first one? It doesn't look like there's anything wrong with that. There's something wrong with it. Look at point 3. What does point 3 say? No colouring or shading. What have they done wrong with the animal cell? So you should be able to see they've coloured the nucleus in. You're not allowed to colour in any structures. So the onion cell's been done really well. The person has drawn the cell wall, cell membrane not touching each other. You have to leave the nucleus empty, like just a black circle. Their arrows aren't touching, but they've forgotten something that was on the other 2 cells. Did anyone spot what they've missed out? And in the third one, can you spot what piece of information that you're supposed to put? You guys, you're right, they shaded the nucleus. They shouldn't have done that. What about the third one? What bit of information have they missed out? That's right, George, the magnification. So they were supposed to put, you have to put the magnification. All right, has this come up before? It's come up as like a 1 marker. Yeah, I don't know if it's ever going to come up as a 3 or 4 marker, but it's best to have gone through it. So there's the mistakes. Don't cross lines, no shading, and the bottom one didn't have magnification. So you need to know that for Tuesday's exam. Okay, here's a quick question. See if you can do these. It's just a naming the parts. Name 2 pieces of equipment the student could have used to prepare cells to view using a microscope. And then for part B, name part A. And for part C, what's the function of B? just going to get my charger, put that in. Yeah, you need a stain and a pipette. A's the lens. That's right, Sophie. Yeah, A's the lens. It's better to say eyepiece lens or eyepiece. So they call it the eyepiece lens. Now, for B, whether it's the coarse adjustment or fine focus, they both involve letting us see the image, bringing the image into focus. So they both allow us to see the image. And for the stain one, the slide, you could really say use a glass slide. That's the glass thing. It's not a slide that you go down on a playground. A little bit of glass, cover slip, stain, drop of water. So any of those. Right, let me put up the answers. That's a bit small. You could have said slide, cover slip, stain, needle, dropper, scalpel, that's for cutting a thin section, tweezers for holding a piece of onion or something so your fingers don't crush it. Eyepiece or lens, yeah, you can say lens, Sophie, so you were right there. You could just say eyepiece or lens or eyepiece lens. And then part A is focus, for B, focuses the image, allows the cells to be made clearer, improves the resolution. They've said ignore moves the stage up and down because that's not the function, is to bring the image closer to your eye so you can see it. I mean, it does move the stage up and down, but that's not its job. Its job is to actually bring the image into focus. Ignore means you wouldn't lose a mark, though. It just means you wouldn't get the mark. All right, specialised cells. One you've got to know across all exam boards. It's the sperm cell, guys. So you need to know the head contains enzymes in a structure called the acrosome. It kind of headbutts the egg. When in fertilization, when the sperm and egg meet inside the fallopian tubes, the sperm headbutts the egg, opens its head and releases digestive enzymes like protease, amylase, lipase. And they digest the cell wall of the egg, and then the sperm releases its nucleus with its chromosomes into the egg, and they fuse with the chromosomes in the egg. And then the egg seals itself off so no other sperm can get in, and then the egg cell grows into you. So the nucleus is haploid. You need to know what haploid means. And it has a tail for swimming. Sperm do. The egg doesn't have a tail for swimming, but the sperm have a tail for swimming. And anything where you're using a lot of energy, you're going to have mitochondria. So watch out for that. Haploid, by the way, means half the normal number. So haploid is 23 chromosomes. Every other cell in a man has 46 chromosomes. So if they ask you about how many chromosomes are in the cell on my head, ear, nose, any other part of a man's body, it's 46, other than his sperm cells. In a woman, every single cell in her body has 46 chromosomes except for her egg cells in her ovaries. And that's it. That's all you have to remember for that. They're known as haploid cells or gametes. Right, this is a nerve cell. And you've got lots of nerves. It's a relay nerve, but you've got lots of nerve cells in your body. And you need to know that they're long, they're branched and interconnected. So if you're asked what's special about nerve cells, how are they good at their job, you can say branched, interconnected, carry electrical impulses, or they're long. Okay. Got these cells here. Anyone got an idea what these cells are? These pink cells, anyone got an idea what these are? 1 second, just going to go beat up my kids. Just had to beat my kids up. Right. What was I going to say? Uh, yeah. So, wait. What are these cells and how are they adapted to do their job? Anyone got an idea? These are known as muscle cells. Awesome, George. They're in the muscles, known as muscle cells. Why are muscle cells good at their job? Does anyone know? What's special about muscle cells? What can they do? Well, they're long and they can contract, okay? And they have lots of mitochondria for releasing energy. All right. Anyone know what these cells are? You all need to know these cells. What are these cells here? These cells are found in plants. Anyone got an idea what these ones are and why they're good at their job? Can you see this picture of these like yellow cells with the long thing on them? Because I'm not sure whether like it's buffering or like glitching at all. Yeah, they're root cells. They're not. You got another word missing there. They're root something cells. They are found in the roots. Yeah, that's right, Alex. Root hair cells. Well done, George. Yeah. That's right, Georgia. Yeah, well done. Yeah, they're root hair cells. How are they good at their job, guys? You could be asked a 4 marker on this. How are root hair cells good at their job? What structures or adaptations do they have that makes them good at their job? What does having that weird long thing, looks like a big nose, like I've got, what does that do? Gives them a large surface area. Awesome. How does that help them? They got a large surface area. What's their job? How's that linked to their job? Anyone know? How's the large surface area linked to their job? So the large surface area, yeah, helps with osmosis, diffusion, active transport, the movement of substances rapidly into cells. Um, oh, let's get rid of that there. Yeah, active transport absorbs water and minerals. Very good. Now, I don't know why my phone is doing that. Right, I'm using my phone to look at the tracks when I flick between. Otherwise, well, half the time I'm staring off to the side. Okay, right, really good. If they do active transport, like Georgette said, and they have to actively transport mineral ions into the roots. What other organelles would we expect? Any time in an exam they tell you active transport is happening, the cells that do that must have what organelles? What cell structures would you need for active transport? Think of the word active. What if you're at, yeah, George, well done. Mitochondria. Active transport. The moment in an exam you see the word active transport or you're going to write active transport, if it's a 1 marker, it's different. But the moment you're going to write active transport, you've got to think respiration, aerobic respiration, mitochondria, energy. So you're picking up marks. Active transport requires energy from aerobic respiration, lots of mitochondria. So you're looking at lots of marks there, guys. Right. Um, yeah, Sophie made a good point. That's another good point. They might ask you, why do these cells not contain a chloroplast? They're a plant cell. Or they may even ask you, are these a plant cell? And some people will be fooled and think, no, there's no chloroplasts. Why do root hair cells not have chloroplasts? Because they're in the soil, they don't have access to sunlight. So it's a waste of the plant's life energy to build chloroplasts in its roots. There's just no point. If you pull roots out the ground, if you take some carrots out the ground or potatoes, I don't know if you've been in your kitchen ever and there's some potatoes that you haven't used for cooking, they start to go green, and that's because they're now getting sunlight. So they go, okay, let's photosynthesize. Let's start making chlorophyll. But while they're under the ground, they ain't going to bother. Okay. So you only need to point that out if they ask you why do they not have chloroplasts. So large surface area, rapid osmosis, active transport, mineral ions, lots of mitochondria, energy for active transport. Okay, just doing a weird flip here. Where is a chromosome found? What is a chromosome? What is a gene? What does a gene do? Where do we find chromosomes? What is a chromosome? What's a gene? What does a gene do? Know the answer to any of those? Okay. Chromosomes are these X-shaped. They're not always X-shaped. That's only when they're doing mitosis that they become X-shaped. But in this diagram, this is. This is in paper 1 as well. It's in cell biology. And it comes up in paper 2 as well. So very basic in paper 1. Probably won't be asked a lot of. But you do need to know that chromosomes are basically a piece of DNA that's wound up, and a short section, a tiny bit of DNA, is known as a gene, and a gene codes for the characteristics like your eye color, hair color, how many protease enzymes are in your stomach, how many amylase enzymes are in your mouth, every single thousands and tens of thousands of features, the size of your red blood cells, how wide the lumen is of your arteries. That's all controlled by genes. Okay. You also need to know for this paper as well, mum and dad donate 23 chromosomes each to the first cell that you were made of. So you've come from a cell made of 46, 23 from dad's sperm, 23 from mum's egg. They come together to make 46, and then every cell in your body has 46. So that's the diploid number. You may have heard of diploid, haploid. Haploid means half, diploid means double. The normal number is the diploid number, which is 46 in humans. The only cells which don't have that are sex cells or gametes, sperm and egg. Gametes, the same as saying sex cells. So you can see here they're not X's, they're just threads. You have 46 chromosomes like threads. Before we do something called mitosis, they double into X's. So, if I was to ask you these questions, what structure in your cell holds the chromosomes? What would you say? Where would you find chromosomes in your cell? Anyone? Anyone? Okay. You'll find your chromosomes in the nucleus. DNA is inside a chromosome. A short section of DNA, a tiny piece of DNA that codes for one of your characteristics, is called a gene. Genes control your characteristics. How many pairs of chromosomes are in each cell? 23 pairs or 46 chromosomes. And why do we have 2 copies of each chromosome? You get half of your chromosomes from your mother and half from your father. That's why you have 2 copies of each. So you have 2 copies of the chromosome with eye color, gene on it, ear shape, head size, whatever. You get a copy of each gene. So from 23 from mum, 23 from dad. All right. Right, zinging into mitosis. What do you need to know about mitosis? Mitosis is cell division, okay, where you have one parent cell that breaks into 2 daughter cells. Now, looks simple there. You can see there's 3 little red blobs, 3 black dots, and 1 nucleus. Well, how did it become 2 nucleuses, 6 red dots or blobs, and 6 black dots? How did that happen? It had to double first. So mitosis is the splitting bit where the DNA splits, which we're going to look at in a second. But the whole splitting of a cell and the whole life of a cell is called the cell cycle. And every single cell in your body right now is at a different stage of that cell cycle. And it's 3 stages. You need to know the 3 stages. One stage is mitosis, where the chromosomes move to each end of the cell. That's called mitosis. It's also cell division. The other stage, the bit that's in orange here in this circle, is the majority of the cell's life. It's just going about its business. It'll be growing, making new organelles, new ribosomes, new mitochondria, more cytoplasm, extracellular membrane, whatever it's got to do. It will then go to its DNA and its chromosomes and double them. And then in mitosis, it will split. And then the final bit, when it tears off into 2, we call that cytokinesis. So, summarizing cell division for your exam here, guys. The cell cycle, if you're asked about it, has 3 stages. The first is the growth stage. Stage 1, growing. Look what my DNA, those little pink squiggles, those little pink lines, they are chromosomes. Each chromosome is made of 10,000 genes, is a piece of DNA with 10,000 genes. They are doubling. So in the orange part of that circle above, that's the life of a cell, just going round and round. In the orange part of its life, it's just doubling its DNA and getting ready to split. It spends most of its life getting ready to divide into 2 daughters. So it does this by making more organelles, growing proteins, and doubling its DNA. So if you're asked what happens in the growth stage, you can get like 3 marks, but you'll probably be 1 mark that you'll only need to say. So you can just say doubling the DNA or duplicating the DNA. You can say it grows new cell structures or organelles and makes protein if you want. Then in the mitosis stage, the cell, the chromosomes become X's. So if you look here, the chromosomes have become X-shaped. That's something you need to know. During mitosis, they form X's. They then separate the X's and go to each end of the cell. Well, they form X's and line up in the middle. Then they separate and go to each end of the cell. And then the cytoplasm begins to divide. Now we have a final stage that they rarely talk about. And this is when the cytoplasm, it goes like a dumbbell shape and then it finally tears into 2. We call that cytokinesis. Sometimes they blend that in with mitosis and they don't really talk about it as a separate stage. All right, so there are your 3 stages of cell division. The second and third are different, mitosis and cytokinesis. These 2 stages are slightly different in that mitosis is where the chromosomes line up in the middle. They can line up vertically or horizontally, doesn't matter. And then they separate either to these 2 ends, north and south, or to east and west. And that's in mitosis. And then it begins to bud and form. It's like it pinches in the middle and starts to separate. Imagine you're pulling you've got some Play-Doh in your hands and you're pulling the Play-Doh apart. That's the end of mitosis. When the Play-Doh finally snaps and you've got 2 cells, we say that's cytokinesis, when it's torn completely apart. So often they blend the 2 stages together, but just be aware, if they start asking you about 3 stages, they're asking you to include cytokinesis as a separate stage. All right, guys, exam question. What happens in each of the 3 stages of the cell cycle? Drink some water. Five-mark question. So you probably get a mark for naming the different stages. So what happened in the first stage? Anyone remember what happens in the big stage? What happens in most of the cell's life? What's it doing? Yeah, it doubles its organelles and chromosomes. Perfect. You get 2 marks for that, George. You can also say growing, making protein. There's quite a few things you could say in that first stage. Quite a lot's going on. It's just growing. So when you say growing, you can go into more detail and say growing some more mitochondria, growing some more protein, doubling the DNA. What happens in the second stage? What's the second stage called? That's the one you all hear about. What do we call cell division normally? Beginning with M. That's mitosis. And mitosis is that you can then name something that happens. Chromosomes line up in the middle. Chromosomes separate to each end. You can sort of say, there's any other things I described there. Chromosomes form an X shape. You can say that as well. And then the final stage is cytokinesis. The cell cytoplasm tears into two daughter cells. Here are the answers. You can really take any four of these. There's loads of different ones there. And there's more answers than there are actually here that they will give you. Yeah, that's the third stage. Yeah. Sophie, mitosis, that's right. And the third stage is cytokinesis. Yeah, cool. All right, let's move on because I'm doing what I did yesterday and taking far too long. Okay, guys, cell X, cell Y. Cell X and Y have just been made by cell division. Name the type of cell division that made X and Y. So we're going like, you know of two cell divisions. You've got mitosis that we just looked at, where the chromosomes line up in the middle, separate, and you get two daughters. Or you got meiosis. So we have to figure out, is this meiosis or mitosis? So what I need to do is bust out my highlighter pen and make sure I don't miss that bit at the top. It's a root cell of an onion. Is this the sex cells of a plant? No, that's the pollen and the eggs. It's not. So what type of cell division would just a normal cell in an organism do? What's the type of cell division for a normal cell? It's not the sex cells, or it's not making sex cells. This one is mitosis, not meiosis. Okay. How can you remember mitosis, meiosis? Mitosis happens in your toesies. Okay, so it happens all over your body, your fingers, your ears, and in a plant happens everywhere. When you write the word meiosis with an E, think of the E as eggs and the S in meiosis as sperm. Meiosis only happens in eggs and sperm. Obviously in plants they don't have eggs and sperm, they have pollen and ovules, but yeah, that's mitosis. What happens to the genetic material, this means DNA or chromosomes, before the cell divides? Anyone got an idea? Before I split into two, before the cell divides, what's it got to do to its DNA? Genetic material. So that was in the growth stage. It's basically asking you, before it divides, it's going to have to double or duplicate. Duplicate, double, replicate. God, I think I went a bit overboard with the questions. All right. Name the two types of cell division in A and B. This one's pretty difficult. If you look at cell division A, this is a really hard question, but let's zing through it. The diagram shows two patterns of cell division. Cell division type A is used in gamete formation. Oh, they've used the word gamete formation. Cell division type B is normal growth. So, what is the division in A and the division in B? So, yeah, mitosis is the second stage. That's right. Yeah, you would get a mark for that. Here, there's a way you can spot which is which. Anyone can see this? Does anyone know which of these is meiosis and which one is mitosis? One of these divisions is mitosis and one is meiosis. So the top division here, cell division A, look how many daughter cells there are. 1, 2, 3, 4. And when this cell number 2 divides, it divides twice to make 4 daughter cells. So that means it's meiosis. And look, there's egg and there's sperm. So they were made by meiosis. Remember I said meiosis, the E is for eggs, the S is for sperm. Eggs and sperm are made in meiosis. So cell division A is meiosis. Cell division B, cell 11 here, splits. That's a zygote, by the way. It's made from the egg and sperm. That's the first cell you were made of. That then divides, and it divides into 2. And then those 2 cells divide into 4. So they're dividing by mitosis. So cell division B is mitosis. No, A is meiosis, B is mitosis. Everyone finds this confusing. So look here. Type A makes 4 daughter cells. Meiosis, you get 4 daughter cells. Mitosis, you get 2 daughter cells. So cell division B is mitosis, 2 identical daughter cells. Name the process. What do we call it when an egg and sperm join? What do we call it when the egg and sperm join together? Yeah, Alex, fertilization. And cell 1, this is difficult. Cell 1 contains 46 chromosomes over here. How many chromosomes will there be in cell 10 and 14? Well, cell 10 is an egg cell, guys, so don't let all the other stuff confuse you. How many chromosomes are in an egg cell? Cell 10 is an egg cell. How many chromosomes are in an egg cell? It tells us cell 1's got 46, but how many would be in an egg cell? 23, that's right, Imi. 23, yep, SM, 23. Right, cell 14 is being made by mitosis, right? How many cells are in 14? Cell 14. Now, I can now see why some of you thought that the second cell division was meiosis, because you looked at those 4 cells, 14, 15, 16, 17, and thought, oh, that's 4 daughters. They're dividing each time. So cell 12 became like 14 and 15, and cell 13 split into 16 and 17. So they're just splitting into 2 each time. There's only 2 daughter cells each time. Anyway, cell 14 is going to have 46 because it's made by mitosis. When a cell divides by mitosis, the number doesn't change. Right, moving on. Got to get through a lot, and I'm running over time. Oh dear. It's because I waffle too much. Stem cells. Anyone know what a stem cell is? If they ask you in your exam on Tuesday, what is a stem cell? What are you going to say? What's the key word for stem cells? Or what can they do? A stem cell is a something cell, or can become something. Well done, Sophie. Undifferentiated. IBH, so it can become specialised. Absolutely, yeah. It's unspecialised or undifferentiated, and you can use the word to say it can become specialised or differentiated. And that's what's happening to these stem cells. They can become muscle cells, nerve cells, blood cells, intestine cells. The last time you had these stem cells was when you were an embryo inside your mum. You don't have those anymore. You've got some really basic stem cells. So if you scratch your skin, you've got some basic ones which will make new skin cells, but they can't grow you a new heart. You've got some basic stem cells in your bone that can make some more red. When you cut and you lose some blood cells, they can make some new red blood, some new white blood for you when you're ill. But you can't grow, you can't chop off someone's hand, you can't grow a new hand, you can't grow a new ear. when you're an embryo, you could chop the embryo in two and it will grow into two twins. So the last time you were pure stem cells was then. Okay, so stem cells are unspecialized. They can specialize into any type of cell. Really interesting medical treatments. They could cure paralysis. They could cure lots of diseases. They could give us organs for heart transplants. But some people believe it's unethical because they say if we chop up an embryo, which is where we need to get these stem cells from, that if we chop up an embryo, we're killing potential life. So some people are against it. You can say unethical reasons for destroying embryos. Yeah. There are also some concerns. If I need a new heart and someone grows one from an embryo, they're worried that a virus, which are very, very small, could get into those cells. And then when they put the organ in me, there's a virus living now that's now in me. So some people say they're worried about viruses being transferred with stem cell treatment. So that's one of the things people are worried about. Here's some questions on stem cells. No, there's no practical chopping the embryo in two. And it wouldn't be infinite baby glitch because it's only got a limited size. Once the embryo gets to a certain size, it's only made of a few hundred cells. And once the embryo gets to a certain size, the cells start specializing, and then you've lost that ability. They start becoming like heart cells, brain cells, nervous tissue. There's a lot of rich celebrities that are getting stem cell treatment at the moment. They're having stem cells injected to keep them looking young. So a lot of the rich celebrities are doing that. They're paying money every year to have like stem cells injected into their face, stuff like that they get from embryos and stuff. It's a big thing with celebrities trying to stay young forever. So what word do we use when a stem cell specializes? We can say differentiates. We can actually say specializes. Name the two types of animal stem cells. Right, you can find stem cells in animals in the embryo and your bone marrow. You've still got stem cells right now. If you've still got stem cells in your bone marrow that make new blood cells for you. So the answers you need to know for GCSE are bone marrow and embryo. Where do we find adult stem cells? Oh yeah, bone marrow. Said that. Which type of animal stem cell can differentiate into any type? That's the embryo, can become anything. Why is the medical profession interested in stem cells? Cure diseases, you can say, grow new organs, cure paralysis. What do we call the type of cloning that produces stem cells that contain the patient's own genes? So, have you heard of therapeutic cloning? You should know about something called... You should know something called this, therapeutic cloning. Okay, therapeutic cloning is where they take one of my skin cells, they cut out the nucleus with my 46 chromosomes, and they stick it, they cut out an embryo cell, chuck away its DNA, and stick my DNA in the embryo cell. That embryo is still an embryo cell. It still can become any cell, but now it contains my DNA. So if they grow a heart from it now, it's got my DNA, and my white blood cells are less likely to reject it and attack it. My immune system is less likely to attack it. So stem cells are a way of growing organs that are less likely to suffer rejection. Main concerns with stem cells is ethical ones, and that they could carry viruses, and that we don't know the consequences of using stem cells yet. Where do we find plant stem cells? Anyone know where we find plant stem cells? Whereabouts in a plant? I ain't got any plants. I used to have a load in there. Anyway, whereabouts in a plant do we find the stem cells? You find the gametes in the pollen, George. You'd find the gametes there. The stem cells is where does a plant grow from? When a plant grows, what bits of the plant grow? That's where the stem cells are. Roots and shoots. Well done, Sophie. So it's the tip of the root, specifically tip, T-I-P, of the root, tip of the shoot. Those are the only bits that grow. If you go to someone's house, and it's not something that people your age do, but if you go to someone's house, you go, oh, what a lovely walnut tree, or what a lovely cherry tree you've got in your garden. You can cut the tip of a branch off, or a tip of a root, take it home, plant it in your garden, and it will grow into an identical cherry tree. So that's what people do. They take cuttings. But if you just cut any old bit, it's not going to grow. You can't just take a leaf off and think you're going to grow a new plant from it. So it's like plants, they maintain like embryo stem cells that can become anything in their tip and their roots. And that's called taking cuttings. You can grow identical plants. That type of tissue begins with an M. Does anyone know what we call the tip of the roots and shoots? This could come up in your exam, a one marker. Begins with M. Anyone know? Meristem tissue. Has anyone heard of meristem tissue? The tip of the roots and shoots are known as meristem tissue. That could come up as a one marker. And how stem cell cloning can help with farming. Let's imagine you're a strawberry farmer, right? I bet that's what you've always wanted to be, growing strawberries. You could take the best strawberry plant on your farm and take cuttings from it and clone it. So you can use the stem cells from the tips of the shoots and roots to grow a field, all of the best strawberries, and get rid of the other ones. So that's what's good about farming. They can increase crop yields and stuff like that. And you can produce insect-resistant crops and things. So these are the answers. I'll leave them up for a second. You can always rewind this if you want to watch it later and go through and have a go at the questions and answers and then cut out all the rubbish bits and test yourself. So you do have to know all of these for Tuesday's exam. So all of these you've got to know. So you can go through later, watch it, just forward through all the bits, rewind through the bits or whatever. Have a go at the questions. Right, need to get a move on. Yeah, you could. It's illegal at the moment to do baby farming, chopping up embryos and making loads and loads of identical babies. But in theory, yeah, it could be going on. Who knows what rich people get up to, or crazy people. Right, diffusion. You need to know the definition of diffusion, guys. You should all know it by now. The one thing that you probably need to be reminded about is to say net movement, which means overall movement. Particles move in both directions. So if you spray some perfume on, some of the perfume moves towards your arm where you just sprayed it, but most of it moves towards a low concentration, so moves away. It's the overall movement, you can say overall or net movement of particles from a region of high to a region of low. If you don't want to say high to low, you can say down a gradient. Down a concentration gradient means from high to low. And it's passive. It doesn't require any energy from respiration. It just uses the natural energy the particles have. Okay, this is a diagram for osmosis. Osmosis is diffusion for water particles, but it can only happen across a cell membrane. Whereas diffusion, if you fart in the room now, your fart's going to diffuse, and if you open the window, it's going to diffuse across the rest of the world and spread, keep spreading and spreading and spreading from high to low. If you spray some perfume, it doesn't need to cross a cell membrane. Water, we only call it diffusion when water moves across a cell membrane. Sorry, we only call it osmosis when water goes across the cell membrane. And it's again the net movement of water particles from a high concentration to a low, but we have to add in across a cell membrane. And it's just for water. All right, core practical that all you guys have to know. Osmosis practical. So 5 concentrations of sugar or salt. You can stick any vegetable in here. Carrot, you could get carrot on Tuesday. They could give you onion. They could do eggshell. They've done eggshell in the past. It can be any material that's made of cells. So anything made of cells can go in those solutions. What happens is there are cell membranes and water can go into the tissue, the potato or the carrot. This is potato, by the way. Water can go into the potato chips and make them swell up, or water can come out of the potato chips and make them shrink. So what you're doing on an osmosis practical, you're trying to find what the real concentration of the potato is. So I've got 5 pieces of potato here, and I've put one in 0.2 sugar, one in 0.4 sugar, one in 0.6 sugar, one in 0.8, and one in 1 molar sugar. I could use that as salt. It could be 0.2 salt. I could be trying to find the salt concentration or the concentration of magnesium. It could be anything, but we can do that using this osmosis practical. Well, what's going to happen is these are either going to swell up, shrink, or stay the same size. So, and then from that, you'll be asked questions. You need to make sure you give them the same. You're changing the concentration of the solution. You'll always change the concentration. You've got to keep the volume. Look, the amount is the same in each tube. Okay, and you want to cut the potatoes to the same size. Now let's have a look at this page. If I put my potato in 0.2 sugar, now they don't have to say 0.2. Why 0.2? That's a molar concentration. They could give you a percentage like 10% sugar. If I put it in there, why did the vegetable swell up? I put vegetable because it could be a carrot, could be a potato, an onion on Tuesday. It could be anything. Okay. Why did it swell up? All you've got to do is say, right, if it swelled up, what happened? What went into it and by what process? So what do you reckon? If I see my potato, I leave it for a day, I pull it out the solution and it's got fatter, tell me guys, what's happened? And this will be 3 marks. Could be, it has been 4 in some of the recent exams for just answering what happened at 0.2. You're right, Sophie. What did the water do? You're on the right track. What did the water do? Why has the potato, carrot or onion or beetroot or whatever it is swollen up? Water moved by osmosis. Boom. Did it go into the vegetable or out of the vegetable in the first one? Awesome, IBH. Water moves into the potato by osmosis. So you've now given direction and process. Water goes in by osmosis. Finish it off. Osmosis happens across a membrane. So, and you're gonna pick up the marks. Happens across a cell membrane, or you can say partially permeable membrane if you really want to. Right. What happened with so cool. Yeah. What I forgot to put there is concentration. If you want to say water moved in from a high to low concentration, you need to make sure you say that as well. So you're telling them that there was higher water outside than inside the potato. All right, at 0.4, what happened? And what happened at 0.6? And again, this could come up on Tuesday. They might say, at 0.4, explain why there was no increase in the mass of the carrot. So why would there be no increase in the mass of the carrot at 0.4? And our answer is, there's no net movement. The same amount of water went out as went in. Why would the same amount of water go out as in? Anyone got an answer for me there? Why if the potato or carrot or vegetable hasn't increased after 24 hours, what does it tell you about the concentration of sugar inside the potato and in the solution? If you leave a potato in water and it doesn't get fatter, what does it tell you about the concentrations inside and outside the potato? Awesome. Nice one, IBH. The concentration of the water was the same outside and inside the carrot. And this means that the concentration of the sugar is the same outside and inside the carrot, which means we've now found the concentration of the sugar. This is very rare that it just so happens to be one of the ones you're testing. This basically won't happen. It would normally be like 0.258 or 0.467, and you have to plot a graph, which I'll show you. And with the final one, so we'd say no net movement by osmosis, water in and out. And with the final one, we would just say net movement of water out by osmosis from high to low, or you can say down the concentration gradient, into the solution across a cell membrane. Make sure you say where it's high, where it's low, the direction, and across a cell membrane. You're going to pick up all the marks, even if you're really struggling to understand. The questions are very hard. They're going to have a lot of data and you're going to be baffled. So you've got to keep to the points. Has it gone bigger? Then water went in by osmosis across a membrane. Has it shrunk? Then water's gone out by osmosis from high to low across a membrane. Why do they might ask you, hey, why did we use percentage change? Okay, so that's a really key point. Why use percentage change in mass? The answer is, and they also, on Tuesday, they might give you grams before, grams up, the weight of the mass of the potato in grams before, 10 grams before, mass of the potato after 24 hours, 15 grams. They might say calculate percentage mass. So you do the difference over the original times 100 to get the increase. Okay, why do we change it to percentage mass? The idea is that although you've cut your chips or your carrots to the exact same size, you can't control the mass. Each carrot chip will have a different starting mass because there's a different amount of cells and sugars in those plants. So you can't cut two identical chips and think that they're going to weigh the same, because that's not the way it works. Okay, so they will all have different starting masses, so it's fairer and easier to compare by changing it to percentage. So make sure you're aware, different starting masses, we have to do it as percentage change to make it fair. Right, this is the type of graph you could be confronted with or be asked to plot on. In this year they asked people to plot this one. So what you're doing here is you're going to plot this up here, percentage change all up the side there, and then you're going to plot your results on. Make sure you use a sharp pencil. The AQA examiners have said, if you're one square out, they're not giving you the mark. So if your pencil's blunt, you've lost that mark straight away. Because if the examiner's like, I don't know which square that's on, like my crappy ones, they're not giving you the mark. So you need to make sure your pencil is sharp and the points are on the exact right place. Okay. Once you've done this, you draw a line. So you plot your points, negative points where the potato has shrunk, water's moved out, and the positive points where the potato has swollen up. And then this point here at 0.4, the potato has barely increased. So we're really close to the real concentration of the potato. They're going to ask you, use your graph to find the real mass of the potato, the real concentration of sugar in the potato. So you'd look there, circle it, that's like 0.42 or 0.43. You circle the line where it crosses 0. If they ask you about this and don't give you a graph, say how would you find it, then you can say plot a graph of change in mass against sugar concentration. Where the line crosses 0 change in mass, that's the real concentration. Right, let's zing on. I want to get done in the next 5 minutes because I have gone way over. All right. Big multicellular organisms have exchange surface areas like alveoli. You need to know 3 things that make a good exchange surface. Alveoli in your lungs exchange oxygen and carbon dioxide with your blood. What are the 3 things, guys? Can you remember 3 things that all exchange surfaces have that are good? Or help efficient exchange. Boom, with the large surface area. Well done, George, IBH. 3 things, not just large surface area. Single cell thick, awesome, Alex. Yeah, they got to maintain a high concentration gradient, and they do that by having a good blood supply. Yeah, so good blood supply, surface area, 1 cell thick, or you can just say thin walls if you can't remember 1 cell thick. You can say both, thin walls, 1 cell thick, if you like. What are these? These are the villi. Villi are in your intestines. Okay, so all of your intestines have got wiggly like fingers sticking out. These are called villi, and each one of the wiggly fingers has microvilli around it, so even larger surface area. exactly the same answer as alveoli. The only difference is they're not exchanging oxygen and carbon dioxide, they're exchanging amino acids, glucose, with your blood. They have good blood supply, thin walls, large surface area. So same thing, guys. Fish gills. What I'm doing here is showing you they could ask you something messed up, right? Could have used the rude word there. They could ask you something messed up and be like, you'd be like, oh my God, I've never learned about fish gills. My teachers haven't taught me this. It's not fair. It's an exchange surface. Every exchange surface has thin walls, large surface area, good blood supply. So keep cool. And they could say it's a frog, explain how the frog skin is good at exchanging oxygen with the environment. You'd be like, frog skin? I've never heard about you just thin, large surface area, and a good blood supply. So same thing every time. So don't panic, guys. Yeah, so don't panic. It's exactly the same every time. If you're doing exchange surfaces, they all have thin walls. Now you need to know the explanation for real extra marks. If it's a 6 marker, thin walls make a short diffusion pathway. Sophie said the good blood supply gives us a high concentration gradient, and the large surface area just allows rapid diffusion, osmosis, active transport. And we're right near the end now. This is just showing you a 6 marker. So this 6 marker said explain how villi and the alveoli are adapted to absorb molecules into the bloodstream. So it's all the answers you've said. So you'd need to say the 3 things that they all have. They both have those 3 things. And then explain how they help. So when you're practicing this, what are the 3 things that all good surfaces have? Alex has already said them there. Thin walls, single cell thick, high surface area, good blood supply. But then if you want the full 6 marks, you've got to explain each one. Why thin walls? Makes a short distance for materials to diffuse. Why folded and all these villi, all these like folds? Large surface area, rapid diffusion. Why a good blood supply? Well, the good blood supply takes away the material as it crosses, maintaining a really steep concentration gradient so that stuff just happens fast. So these are the explanations. Large surface area gives rapid diffusion, good blood supply maintains concentration gradient, thin walls, short diffusion distance. One extra thing that's always an extra mark is if you mention the fact that villi have an extra villi sticking all around them called microvilli that gives an even larger surface area, and alveoli, villi and fish gills would have mitochondria, or frog skin or whatever they give you, because if you're exchanging materials like your kidneys, or your lungs or your liver, you're going to do active transport, or your intestines as well, or the roots of plants, and so you'll need mitochondria for that to make energy. Right, very finally, we're going to finish in 1 minute. You need to know this off by heart because I've run out of time, but can you name the cells and tissues in the structure of a leaf? They're up here. You need to kind of know the top is the upper epidermis, the middle is palisade mesophyll, then you've got spongy mesophyll, then vascular bundle, xylem, phloem, and then the lower epidermis is at the bottom. And you need to know how they're adapted to help with photosynthesis. So they could ask you a 3 marker, 4 marker, or a 6 marker. So all the details there, I'm not going to go through all of them, but yeah. The upper epidermis is see-through, so light can go through. The palisade cells or tissues are packed with chloroplasts for photosynthesis. They absorb light. Spongy cells have air gaps to allow diffusion. And the vascular bundle, the phloem, carries sugars, the xylem, water and minerals. And the lower epidermis contains stomata to allow carbon dioxide in for photosynthesis. And the guard cells open and close the stomata. So you can revise that. That's on here. You can rewatch and revise that. There's a 6 marker here, which this is a 6 marker. I've cut the bottom off of it, unfortunately, but you can revise that and then check the answers after. You could like pause it, have a go at it. These are some of the answers. It's just basically going back to this page and naming explaining what each structure does, so you need to know that. And that's that. Went a bit over what I wanted to do. So tomorrow, going to cover. I'm going to put a time up. I'm going to cover like heart, lungs, circulation, and other things as well. I keep waffling on for too long. So thanks, guys. Take care, and yeah. These are up on for you to look through again if you need to, but they're a bit long for that. I've got loads and loads of videos on my channel as well you can look through that revise lots of other topics as well in science, and they're organised into playlists, so for your exam board and stuff like that. So nice. Yeah, going to do organisation tomorrow, I think. All right, guys, take care. I will be doing one for chemistry. When after the biology exam, I'll start doing some for chemistry. I'm going to make them a bit slicker. Like, obviously, I've just been kind of banging these out to try and get through. Just get the information over. But yeah, good luck, and no problems at all. We'll do. Take care, guys. All right, nice one.